All right, so thanks so much for your attention. I guess two disclosures to start. One is I packed way too much stuff into the presentation, so for those of you who stuck around, I'm going to talk very quickly. The other is that this talk could not be less like the previous talk, so for those of you who stayed expecting more of the same, it's going to be very different. My talk's divided into two sections. First, I'll talk about our studies, how we're trying to put genomic medicine into practice at our hospital and our research institute. And second, a number of specific examples and results and lessons learned from this experience. I guess I should also apologize, I'm pretty sweaty because I ran on the treadmill for the folks from Greenwood, so sorry if I'm dripping and covered in sweat. So to start, to put things in context, we're located outside DC in Northern Virginia. We're a decent-sized healthcare system. We have about 6 or 7 main hospitals, a large network of outpatient centers. And we focus on pediatric and neonatal and fetal genomic medicine. So the good news is that we have a lot of deliveries, about 20,000 deliveries system-wide. About half of those are at our main hospital where our research institute is located. And our institute was started about 5 or 4 years ago, depending on what you count as the starting point. And the idea was to do relatively large-scale genomic studies, understand health and disease through these studies, and I think just as challengingly, figure out how to put this information into a working healthcare system to take better care of our patients. So the themes of our studies include we do trio-based whole genome sequencing on pretty much all enrollees, and so that means mom, dad, and baby. We generate as much other omics as our samples allow. We have a very wide biobank where we get everything from placenta to tissues affected with certain conditions, to blood, to saliva, anything you can imagine. We get everything from our electronic healthcare record system and supplement this with study-specific surveys, and we bring this all into one database to help with statistical power regardless of the specific question being asked. So this is a very simple slide, obviously, but this gives you a sense of where our genomes are coming from. This is the number of genomes we have with matched clinical data. As you can see, most of these genomes are coming from a longitudinal study. So this is a study where we have research nurses embedded in a number of OB practices. Patients are enrolled about halfway through pregnancy. We do whole genome sequencing right when the babies are delivered, and we follow them longitudinally, hopefully for their entire lives. And whether they immediately have a congenital condition that's identified within the first couple of days, or if they develop asthma or autism later, we can do whatever analyses are called for. We also, as you can see in green and blue there, we have some condition-specific studies as well, and I'll talk more about the results of those later. I know this is hard to see, but all those numbers in the map is showing you where the genomes of participants in our studies are from. So about 2,500 of those are babies that were born in Virginia. The rest are the parents of those babies who we sequenced as well, born all over the world. So a very ancestrally diverse cohort. And then this is just a simple PCA plot, of course, on the other side showing genomically where about the first 700 adults in our studies are from. I know for this audience it goes without saying, but having the inheritance information about all our participants and then having the ancestral information helps us, we think, more intelligently consider the pathogenicity, the chance that a given variant is disease-causing in a given individual. And then I know a lot of us are the penguins on the iceberg, right? We're the folks that are interacting with the clinicians and the patients. But naturally, much more important, or at least as important as folks like me, is this very robust IT and informatics infrastructure below the surface, right? That needs to be running very smoothly to aggregate the data, to manage the data, to secure the data, to allow our collaborators and clinicians into the data. And it's a big project and one we devote a ton of energy to. So I'm going to shift gears at this point. I know I apologize, this is kind of a trite graphic, but the idea is I'm going to start with some of our simpler analyses and results and move on to slightly more complex there. If anybody has a better example I should use for this, please send it to me. So we'll start relatively simple, and these have to do with a lot of these, N of 1 or rare disorder analyses, and these are just images from babies in our NICU that we've solved through trio-based whole genome sequencing. We're able to achieve results in about 60% of cases, and this is again where clinical sequencing and clinical testing didn't arrive at an answer. We get these patients through a specific study devoted to this, but we also get these through our longitudinal study. So we take all comers for our longitudinal study. As shown in blue there, most of the folks who participate in these studies are healthy, fortunately. But as shown in all these other colors, we get the expected proportion of these Mendelian or kind of rare disorders individually that we can dive into on an N of 1 basis and provide answers for these folks. And I'll brag a little bit, we're very successful at this and about doing this efficiently. I know this is a completely uninformative graph, so let me explain this. When we try to aggregate some of the results, we're able to compare things with standard testing. So this is from a study we did on about 1,700 infants, comparing standard newborn screening, from the heel stick that gets sent off to the state lab with whole genome sequencing-based screening. So on the x-axis, each of those tick marks is a different gene of the 163 genes that correspond with newborn screening conditions. On the y-axis is the number there. So in red, these are the number of mutations in these 1,700 babies in each of these genes that are found in ClinVar or HGMD and are annotated as pathogenic or disease-causing. In blue are those as well as the ones that are novel but look like they're disease-causing. So a novel truncating mutation in these genes or something else, a whole gene deletion and that kind of thing. And so the bottom line is that there's a lot of data in there that aren't represented in any of the databases. One of the things that we feel is our responsibility is to help the community. So we've made these data, all our genomes, available through a partnership with QIAGEN that research labs and biotech groups can get into so that they can use us as a reference database for both phenotypic as well as genotypic data. One of the things that's talked a lot about, as you guys know, with whole genome sequencing and newborn screening is that we can extend the range of conditions that we look at. So this was a baby that was in our NICU, as you can see, with massive hepatosplenomegaly, normal newborn screening. At first they thought it was some kind of anatomic thing, but through whole genome sequencing we found that this kid had biallelic mutations in NPC1. And so we were able to figure out this result. We were at a conference much like this in California when we got this result and immediately validate this, return the results, and get this patient in with the world's experts on this condition. So he was able to get medications to hold him over to liver transplant, which he's had now. And now that he's post-liver transplant, he can get the same medication intrathecally for neuroprotection and to hopefully help with his cognitive outcome. I think less scientifically interesting, but more common, we have a lot of kids in our cohort, about 15 so far, who have G6PD deficiency, right? So this is the most common enzymatic deficiency in humans. Kids are usually okay until they get exposed to certain medications, certain foods, or get infections, and then they can go into sometimes life-threatening hemolytic crises. So we were able to find these results and return them. This is a condition that's screened for in newborn screening just north of us in DC and in Pennsylvania, but not in Virginia where we are. So not as scientifically exciting, but certainly impactful on these kids' lives. So I'm going to shift gears to the more complex now. This is looking at the molecular study that we have on preterm birth. If you look just at the clinical data, the area under the curve maxes out at about 65%, meaning, and this was done in a collaboration with the group GNS based here in MIT, or an offshoot of MIT, I should say, who have done great work on this. And so in other words, you can predict if someone's going to have a preterm birth with about 65% accuracy just from clinical data, which is okay, but not great. We did a traditional GWAS style analysis, and we got some hits here, but I think we're statistically underpowered to get obvious hits from the genomic data alone. So next, in collaboration with folks at ISB, the Institute for Systems Biology out in Seattle, we've done analyses where we've now combined the genomic data and the clinical data. And so this is a random forest analysis showing in red, those red circles are preterm deliveries, the blue circles are term deliveries, and the orange are kind of late preterm deliveries. And so you're seeing through this, through the clinical and the whole genome sequence data, some separation, slightly better ability to predict who's going to have preterm birth. Again, in collaboration with the guys at GNS and women at GNS, we've done now studies looking at the clinical data, the whole genome sequence data, and from mom, the epigenetic data and the transcriptome data. And so each of these circles represents a different one of those variables. We're only showing you the biggest hits out of the millions and millions and millions of data points. The bigger the circle, the more significant. The blue ones are the most significant ones, and these are the ones we're now validating in separate cohorts. And that if they validate, and the things look promising so far, we'll be able to pull off into a biomarker assay to do for a woman that's going to become pregnant or has just become pregnant. And we're doing some traditional proteomic assays. So we're doing a study on our placental biobank where we're comparing preterm placentas to term placentas and finding biomarkers and now validating those in the blood samples of the moms from about 20 weeks delivery who have participated in our studies. They get these blood samples as, for diabetic screening. We're also doing some work, some slightly more basic work looking at correlations with de novo mutations. So we're looking at why certain folks have more de novo mutations and where they tend to occur. And because of the size of our cohort, are able to find some significant results there. And so with that, I'd like to conclude with a few lessons learned. First, I think for this audience, you guys know it's really all about bioinformatics now, right? So we're moved away from this scenario where a patient comes in, we order a test, it's wrong, we scratch our head, we order another test, it's still wrong, we squabble and do all sorts of crazy, have affairs, and then we finally get the answer. So now when the patient comes in, everybody looks up when I say that. So now when the patient comes in, we try to generate as much omic data as possible, and the job, like me, a clinical geneticist, is to interpret these data at that point, rather than to just say, oh, let's do this test and let's do this test in a piecemeal or kind of shotgun approach. The second thing is I know that all of us are excited about this idea of personalized or precision medicine, and I won't ask where you guys think we are in this graph of hype or whatever. But I think the bottom line, the thing I try to impress upon patients that I see and clinicians I interact with, is that we're really at the early stage here in understanding what a single variant does to impact human health, let alone variants in concert, let alone variants with non-genetic information and so on. And so maybe this is just kind of my plea for job security, but there's certainly a lot of work to do to understand how this impacts human health. And along those lines, we all understand that this is very complex information, and the medical system is already, I don't want to say quite at the breaking point with the complexity as it is, but it would not be helpful to simply add ridiculous amounts of data to what they're already trying to deal with. So one of the things our institute works very hard at is trying to package this data and return it to clinicians, return it to patients in a way that they can take advantage of our expertise. So a very simple thing is we've developed an app that folks can chat with me and my team and genetic counselors on our team and postdocs and bioinformaticists about any question they have about genetics and genomics, whether it's research or clinical based. And I think honestly, the exciting thing for us is doing these sophisticated genomic analyses, but at the end of the day, this is often just as impactful as some of the stuff that all of us wake up for excited to do every morning. And then finally, I hope you'll agree that it's just an amazing time to be a geneticist or genomicist or whatever you want to call us these days, with the kind of data that we can gather, with the kind of hypotheses we can address, and with the kind of impact we can make on patients and their families. And with that, I'd like to thank you guys and all the folks that work with us, all the folks at ITMI who have braved this and have supported all of us through this work. Thanks so much, guys.